This question is from the chapter Biotechnology, Principles and Processes of class 12th textbook. Question 111, NEET 2023. During the purification process for recombinant DNA technology, addition of chilled ethanol precipitates out. See, we are isolating DNA. So chilled ethanol will precipitate out DNA. The answer is DNA. This is a long step and different questions can be asked on this topic. So I will give an explanation of all the steps. What are the steps and why those steps are needed. So DNA isolation step. So this is the cell. Usually plant cell or fungal cell or bacterial cell is used. So these cells will have a cell wall around the cell. So first we have to get rid of the cell wall. Next step would be to get rid of the membrane. Next, this is the nucleus. So nuclear membrane will also be digested simultaneously when the cell membrane is dissolved. And then the all the cell content will come in the test tube. Then we will have to get rid of the proteins present in the cytoplasm in the lysate, remove the contaminating RNA also, and then finally we will get DNA. So these four steps are essential to finally reach to the level of DNA. So cell wall, supposing if I am using plant cell, then the enzyme I will use is cellulase because the cell wall is made up of cellulose. So cellulase is going to digest the cellulose. If it is a fungal cell, then chitinase. Chitinase digests the chitin. Next, if it is bacterial cell, the enzyme would be lysozyme. Lysozyme acts on peptidoglycan, which is the component of bacterial cell. So we have destroyed the cell wall. Next is the membrane. For membrane, there are specific detergents. Those are called molecular biology grade detergents. So detergents are going to solubilize the membrane. So the membrane, the cell membrane as well as nuclear membrane will open up. So then you will have a lysate which will have a mixture of protein, RNA and DNA. So the lysate is then treated with proteases to digest all the contaminating protein. To digest RNA, the enzyme is called RNase. So contaminating RNA will also be digested. So what is left in the lysate in the solution is DNA. Now it is invisible at this stage. To precipitate out, this is what the question has asked also. I'll change the color now. So the question is asking what is used to chilled ethanol is going to precipitate out. What? So the DNA which is in the solution will be precipitated out using chilled ethanol. Because DNA is negatively charged, it is polar molecule, and the charged molecule will come out in the solution if due to excess of chilled ethanol, and then with using a glass rod it is removed. So I hope I have given clarity on this, all the steps and why it is done, what is used and why it is used. Thank you so much for watching this video.